Chapter 11, Nucleic Acids and Protein Synthesis, Part 2, with our continued commentary, our misspelled continued commentary. In this part, we're going to talk about the translation process. Yes, it's infinitely more complicated than what you see in the slides. So for this intro level class, we've simply summarized the key points. So make sure you memorize all of the three different stages or parts of the translation process. Now, I have a quick summary and I have a video that goes along with the quick summary. I also have a more in-depth summary which you'll be responsible for. So let me start off with a quick summary. There is a video that goes along with this, and I've basically taken stills from the video to help explain what's happening, just to give you a better context of this process. So let's go ahead and start off and begin. We have three stages or parts. We have the initiation, elongation, and termination. So our goal is to take the messenger RNA and use it as a blueprint in order to construct our proteins. In the first step, initiation, we have a very long single strand which is our messenger RNA. What's going to happen is we're going to have a molecular train, if you will, called the polyribosome. Notice it's made up of two different ribosomes which are coming together like a sandwich around the train track messenger RNA. And when they come together, they're going to ride the messenger train track, so to speak. Now, just as you need supplies for any factory in order to build your product, and you need something to transport the supplies, we have transfer RNA. And transfer RNA is going to transfer in the desired amino acid. Now, key part here, the very beginning of this translation process in the initiation stage, we have what's called a start codon. This signals where the translation process is to begin, and that it is to begin. So we have a special transfer RNA that comes in along with its amino acid. At the same time, we have the polyribosome, and they all come together and they're ready to begin. Elongation is the actual translation process where we start to form amino acids. The orange, green, and blue ugly looking pictures, those are supposed to be amino acids. The polyribosome has two slots. And we'll actually name those slots at a later time. They're actually called sites. But there's two slots that the transfer RNA truckers can come in. And you need the proper transfer RNA containing the proper amino acid. On the surface of the transfer RNA, which I'm circling right here, we have what's called the anticodon. And the anticodon needs to fit to the proper codon. This is how we know that we have the proper amino acid that is going to be put into the sequence. Now, what happens is, during the elongation process, the polyribosome is like a train that's going to ride the train track of the nucleotide words, so to speak. So here we have them grouped together in their words, and it's going to ride the train track. Now, what happens is we have our old and we have our new one. The old transfer RNA already gave its polypeptide. The new transfer A is the incoming transfer RNA with the brand new amino acid. So, during the elongation process, two things are going to happen. One, the polyribosome is going to ride the train track and move over to the next new word of nucleotides. At the same time, the amino acid is going to form a brand new peptide bond with the growing polypeptide. As the polyribosome moves over, the old transfer RNA, which is no longer connected to the its old amino acid, is going to be ejected. And the new transfer RNA is going to be basically revolved over to the old slot that the old transfer RNA was in. And this will continue on. My advice is to watch the video during this process. The last step is the termination step. This is when the polyribosome finally encounters the last codon responsible for, or the last codon. So the polypeptide has finally been made, or the protein has finally been made. So this last codon we call the stop codon. And we don't go into details, but we do say that it signals a special protein called the termination factor. So this chicken leg looking thing right here is a termination factor, and its job is to basically separate the polyribosome mRNA. So basically its job is to dismantle the ribosome and end the translation process. Here is before, and then here is after, when it's been dismantled. Here is our brand new polypeptide chain that has been formed. Now, we've already talked about hydrophobic packing and all these other things which help maintain its shape. Bottom line is there's all this extra stuff that happens afterwards that you're not responsible for in the translation process. You just need to know what happens in my translation summary step, and that's enough. Bottom line is it produces the polypeptide. So memorize all of this, the termination factor protein, the stop codon, and how it dismantles the ribosome and ends translation. Here is the more in-depth steps now that you have a point of reference of what's roughly going on. So you're responsible for memorizing this. Now, in the initiation stage, we have the large and small subunit which make up the polyribosome. The small one is put in first, however, and then the second one, or the larger, is put in last. When it's first put in, it lines up to the start codon, or the initiating codon, same thing. The initiation sequence, the word is AUG. Now, what happens is, during this small subunit being put on to AUG, the next thing that's going to occur is we're going to have that special transfer RNA. And its special name is called formal methionine. Formal methionine. Well, I take that back. It's formal methionine if it's for prokaryotes. It's methionine if it's for eukaryotes. Bottom line is it's going to be a methionine. So let me correct myself. It's methionine for eukaryotes. It's formal methionine for prokaryotes. So in this case, we're assuming it's going to be a prokaryote because it's F-met, so formal methionine. So here we have our special transfer RNA, formal methionine. Notice the anticodon must match up with the codon of the messenger RNA. It's the same rules where we had hydrogen bonding, and you notice just as A always went with T, well, A always goes with U, and C always goes with G. So this is how the messenger RNA recognizes the proper transfer RNA. So you have the proper amino acids in the sequence. Next thing we have is the larger subunit that comes in. And in the first slot right here, which I'm going to go ahead and highlight, we have two slots. The first slot is going to be called the P slot. We have another slot, and I'll use the red color. This slot over here, which we're about to talk about in the other slide, we call the A site. So first slot is the P site, second slot is the A site. I use the term slots a lot, but the proper term for this is sites. So P site and A site. And you can think of them as giant slots, if you will, that the transfer RNAs can fit in. Kind of like, I always think of it as like a two-barreled gun. A gun of love, which shoots flowers, but nevertheless, two barrels. So in this case, we have elongation in step 2. This is the process where in the first step, you have your old transfer RNA, you have your new transfer RNA, which just came in. The new transfer RNA has an amino acid which is going to connect to the growing polypeptide. The special name for this is peptidyltransferase. Peptidyltransferase. So, yes, you're responsible for knowing the name of the enzyme responsible for peptide bond formation, peptidyltransferase. So here we see it connected. So the old transfer RNA is going to then be ejected as the entire polyribosome shifts over to the next codon. So here is before, and here is after. Notice, as it shifts over, the transfer RNA in the A site, basically the barrel is going to rotate, and it's going to go from the A site to the P site. So in picture C, we have the after. The new transfer RNA is now in the P site, and it now becomes the old transfer RNA. The original old one, which is now empty, is ejected. Now, this process occurs over and over and over. Peptidyltransferase for transferring it, and then as the polyribosome moves, we have translocation. It's transferring to the next messenger RNA codon. So, you're responsible for knowing this, know all the different steps, know the enzymes, and know the actual movement is called translocation when the polyribosome moves. Termination, we talked about termination. When you reach the stop codon, you have the termination factor protein. And it basically releases the brand new completed protein as well as dismantles the rest of the polyribosome. So here's our new completed protein. Here's our dismantled polyribosome and messenger RNA units. So once released, protein folds into this proper secondary, then tertiary structure and quaternary. There is other steps after this. We call this post-translational steps, but we're not going to get into those at this level. So here's an overall summary and a couple other items which I could test you on. Make sure you memorize all of these as well. Any changes resulting in an incorrect base sequence on DNA. So this is the definition of mutation. Let me say that one more time. Mutations are any changes resulting in an incorrect base sequence. So in this case, a good, statistically good acceptable amount of mutations is 1 in 10 billion. We have mutations all the time. Some of them are environmental factors. Some of them are just mechanical errors from the transcription translation process. Even though it's not set on here, we have a ton of special repair enzymes. So there are a ton of enzymes which basically go and proofread our DNA, and we have special enzymes which also help proofread. Well, we won't get into that. So basically, we do have special proofreading DNA repair enzymes. So FYI, that's how we fix all of those errors. If we want to multiply the number of mutations, because we're mean like that, we can use X-rays and UV. Basically, they excite electrons within covalent bonds of the DNA and break and make new bonds. Or we can add in a mutagen. These are any chemical agents which cause mutations. Although not necessarily harmful, I'd say 99.9999% of the mutations are harmful because you're messing up a structure or process that's already there. So it's not like Spider-Man where every time you get a mutation, something good happens. Usually it's something bad like cancer or a genetically related disease such as sickle cell anemia, which is passed down. Oftentimes, however, we like to change the DNA in a lab on purpose if we've found ways to improve upon a piece of DNA. We call this genetic engineering. Any altered DNA that we've done on purpose, we call it recombinant DNA. One of the best examples of genetic engineering that worked and that we used all the time was altering E. coli so that they would produce insulin. So this concludes our video commentary for this section. My advice, make sure you memorize everything in this slide right here. Make sure you memorize all the steps. And above all, as you're trying to understand this process, watch the video so that way you can see it in motion and know all of these steps, as you will see these on the next big exam. This concludes the Chapter 11, Part 2. You'll also see this on any other testing materials, such as quizzes, online quizzes. And let's stop here.